I picked a title that I thought was catchy: Intelligent Design as Idolatry. The argument being that worshipping or viewing us as watchers is a little bit like mistaking God for an image of God. Intelligent design people don't worship watchers, so a friend of mine suggested perhaps I need something like abstract animism. That is mistaking man-made objects for actual organisms. So I'm not going to try to make strong reference to Darwin or natural selection, and one of the reasons why I'm not is that for most of us that are committed to evolution, we take for granted the complexity of organisms. There's variation followed by selection. Variation of what? Variation of complex organisms. So in some sense, Darwinism takes for granted the natural origin of biological complexity. But I've got friends who are seriously concerned about the validity of evolution, who are not extreme religious types, but I think are concerned about where does that complexity come from? How does it arise in the first place? And what I want to do in this lecture is try to explain to you where and why I think that happens. I said this is sort of experimental on my part, so I certainly would appreciate feedback also as to whether these arguments are convincing or at least comprehensible. I'm going to start off with where we are and then move quite quickly to where I'm from. I'm actually Australian, but almost all of my forebears are New Zealanders, and in about 1830 my great-great-grandfather was shipwrecked here and was saved by some local Maoris. He converted to Wesleyanism, Methodism, and became a missionary in this part of New Zealand here. He was the first to translate the Bible into Maori. And then a couple of generations later, my father and his brother were born. And then almost 50 years later, here they are. And I'm using Uncle Dennis as a foil here. Now, Uncle Dennis and I have been emailing extensively over the last several months, because Uncle Dennis spent a lot of his career educated here, spent a lot of his career in the geothermal fields in New Zealand, these rather spectacular areas. And he worked in these geothermal areas, and he did some mathematical modeling that showed that this field wasn't going to generate more electricity, so you shouldn't build more of these plants. He's also the first person to describe a method for describing the rate at which sap flows in trees. So he's a full-blown scientist. He's also a devout Christian, and in correspondence that we've had, it's quite clear that he feels that God can intervene in the natural world. And a year ago, he wrote a piece for his local church, sort of saying that he could destroy his nephew's career at Harvard, but he won't do that because he knows, because the poor nephew, me, doesn't understand how wrong I really am. So this is a one-sided conversation with Uncle Dennis to try to explore the ways in which organisms are not like the mechanical devices as his training as an engineer, as the mathematician might suggest. So here is the proverbial watch. Dawkins talking about the blind watchmaker. Amazingly complex things. And what I'm going to basically argue is the ways in which we are not even vaguely like a watch, even though we are highly complex. Some of the things that are characteristics of watches is that if you look at the parts, there are all these cogs, but they've got different diameters and different size and numbers of teeth. The screws are often different sizes. And one of the hallmarks of watches is that the pieces are not interchangeable. And that's going to be very, very important in this talk. So if we look at the nature of a human design watch, they're built from the top down, from the outside. They have designers. There are relatively few parts compared to us, as I'll explore in a moment. Most of the parts are unique. They're not interchangeable. Once made, it just tends to run down until it no longer functions, sort of entropy or the second law of thermodynamics. And finally, it's virtually impossible to change it so that it functions in another way. It's not really evolvable. I don't know how you take a watch and turn it into a microwave or any other type of device. And I think it's largely because of the relatively few unique parts, which we'll explore. So if you really thought that organisms were like watches, I would have to agree with you that I can't see how evolution would operate, despite the very compelling arguments of Darwin with variation followed by selection. So what I want to do is, starting from that position, what are the ways in which organisms are not like watches, and where does biological complexity come from? I also want to emphasize that part of these arguments I'm going to make depend on almost 2 centuries of science. And there are things that we know now that if you didn't know, I don't see how you could come to believe in evolution. And I want to explore those as well. And so particularly if you start with the naive mind, who might start looking at mechanical devices and compare them to us, let's explore the reasons and the ways in which we're different. There's this really wonderful book called The Way Life Works. It's got nice cartoons in it, and some of the ideas are drawn from that, and I strongly recommend that. I mean, I've almost been tempted to set that as one of the texts from my non-majors course that I teach on dinosaurs, but it's also got sort of biology as a foundation. And in this book, they list sort of 16 things about life that we think we know. It's organized hierarchically over multiple scales. It builds from the bottom up. It needs an inside and an outside. Well, so does a watch. Life uses a few themes to generate many variations. Life organizes with information. Life encourages variety by reshuffling that information. Life creates mistakes. Life occurs in water. I'm going to look at just a few of these in this lecture that I think are particularly relevant to understanding how we're different from a watch, and I'll address each of these individually as we get to them. So you'll re-see these 4 headings as I proceed through the lecture if you can't write them down now in this time. Life runs on sugar. Life works in cycles. Life recycles everything it uses. Life maintains itself by turnover. Life tends to optimize rather than maximize. Life is opportunistic. This one I think is critically important. Life competes within a cooperative framework. And people talk about Darwin, they tend to emphasize the competition, but not within the cooperative framework. And the reason for the cooperative framework is we all rely on each other for resources. We can't just go it alone. And life is interconnected and interdependent, which is related to 15. And I'm going to look specifically at this optimize versus maximize issue. Things we couldn't know if it were not for science that are crucial to my argument. We're made up of parts, cells. These parts interact via universal rules, protocols, and I'll explain that in a moment. It's critically important. These rules are encoded in the DNA that lies within the cells, so that's part of the hierarchy now. We're made of organs, which are made of cells, which has DNA inside it. Life builds from the bottom up. And the DNA itself is made of interchangeable parts that also operate via protocols, and these are sort of the rules of chemistry. So these things I think are crucial to understand the fact that we are able to evolve. And these are things that are not knowable if you don't have access to science and the invisible world that it's revealed within our own biologies and bodies. So I'm going to ask 3 questions now. What enables biological complexity? What drives the complexity? And then what shapes the complexity? So starting off with what enables the complexity. So I want to deal with this issue of protocols and being made up of parts. It's critically important. So here is a pile of rocks. And I think you'll all agree that that's metastable. It ain't going to stay like that for long. If I have more rocks, I get a bigger pile of rocks. And if I pile on even more rocks, I get a bigger pile of rocks. There are no rules of interaction. They just sort of pile on top of each other. On the other hand, we can take a different type of entity, a Lego piece, and Lego pieces have these little nubbins in it and these holes, and they're always equally spaced. These are the protocols. These are the universal rules of interaction. And so, of course, what you can do is with a few pieces make a simple device, and with a lot of pieces you can make bigger devices. So as soon as you have universal, simple rules of interaction, you can build up complexity in a way that you can't with, say, no protocols, like a pile of rocks. And of course, this is relatively stable. So the key thing here is that life operates with these protocols. Cells know how to talk to cells in simple ways, and the instructions lie within the cells, the DNA. So that is to say, life is organized hierarchically over multiple scales. That hierarchical organization is absolutely critical. Watches aren't built hierarchically. There aren't bits within bits within bits. So this is shown from a diagram from the book that I referenced earlier, with atoms, molecules going up to cells, cellular communities, tissues, organisms, ecosystems. And so here, mostly because it's good fun, this is a pin head, a head of a pin, and each of these little dots is a bacterium. It's just interesting that bacteria are only slightly outside of our visual acuity, and of course a picture of a cell showing here a purple should be a purple colour since it's an E. coli, which of course is the source of our mitochondria that produces all of our energy. So of course all of the energy that we use in our cells comes from the bacteria that we've got as permanent residents within our own cells, the mitochondria. So that hierarchical organization is critical. And then of course if you take something like a tongue, there are lots of things like taste organs that are made up of cells, etc. And these are almost invisible to us. And so I think it's important to emphasize that we are sustained by a myriad of invisible processes. And so I think if you're going to teach something like biology, it's worth emphasizing the importance of the invisibility of this, that we can't intuit it by just looking at ourselves. We actually need technical tools to understand that. Life organizes with information. You're all aware of this. I just want to emphasize the amount of information that I think we often underestimate. So here are 3 base pairs of DNA, so the DNA unraveled from a chromosome sitting inside the nucleus within a cell. How much DNA do you actually have? Well, most human cells have 6 billion base pairs, 6 gigabytes, or probably gigaquads since there's 4 nucleotides rather than a 10 byte. Typed out, it would fill about 2,000 volumes the size of the Lord of the Rings. It's about 2 million pages. So you can imagine a series of bookshelves with Lord of the Rings on it, and that's just the DNA from one of your cells typed out. So it's a phenomenal amount of information. It's so huge that it's actually visible to our naked eye. And so here are 3 base pairs now with a model of DNA, and you can see the distance between the base pairs is basically the distance about the size of an atom. So the DNA is packed in at the atomic scale, but you multiply this by 6,000, and it would stand about 6 feet, so taller than I am. So all the DNA at the atomic level in your cell, 6 feet. Now, it's worse than that. We have about 10 trillion cells. 10 trillion cells. If you took all of that DNA, it would go to the sun and back 67 times, or to Pluto and back almost twice. So there is literally an astronomical amount of DNA sitting inside our cells. Typing it out would fill about 10 billion laptop computers, each with a 40 gigabyte hard drive. 10 billion laptop computers. That's the amount of DNA that I've got inside me. An incredible amount of information. And we thought we invented the information age. So if you want to try to understand where this complexity comes from, it makes sense that we have an enormous amount of information inside. We're not made up of a few little trinkets. Well, how does all that information get translated into morphological form? And we're making progress towards that, and I want to try to explore some of the actual facts that we know, but also conceptually the way I think it's important to think about how that information gets converted into us. So Darwin, of course, said that we are profoundly ignorant of the causes of each slight variation, let alone where the animal came from in the first place. He took it for granted. He noticed empirically that there was variation and argued that under the sometimes varying conditions of life we might expect to see if those things are heritable, change through time. However, we do now know that iterative processes, things that grow, with simple rules can lead to complex structures. And so perhaps you've seen these arguments where if you have a simple rule of putting a triangle within a triangle, within a triangle, within a triangle, within a triangle, you can develop simple complexity. And sometimes there are things that look a little bit like that, patterns on a shell. Here's another example here. What I have is parent cells. So there are 3 cells in each of these boxes, and the 3 parent cells determine the state of the daughter cell. So here, if all 3 parent cells are pigmented, so is the daughter. Here, if they're all unpigmented, so is the daughter. So with 3 cells and an on-off, there are 8 possible combinations, 8 rules. So this is the rule set. So if you start off with a couple of cells and then you growth down that way, you can get a pattern, which is pretty dull looking. Now if I just change 2 of these rules, so the output's reverse compared to that one, I get a different pattern. And if I do it again, change 2 more of the rules, I get a different pattern. And if I let that pattern go, something really bizarre happens. So far these are a little bit boring in the sense that if you see this part of it, you've seen the rest of it. But look what happens with this one. Here's a structure appearing here, at a scale length that's much, much larger than the size of an individual cell. The instructions going into it are extraordinarily simple. They're just these set of 8 rules here. And so we now know that you can get the emergence of complex, large form much larger than the individual cells themselves. So this has been a staggering discovery. It's led to notions of chaos, etc. But I think it's critically important. The other thing that's critically important about this example is that when most people think about mutation, it's the damages of things that means the organism doesn't work. Well, here's a case where we have coherent mutation that changes the form of the pattern of cells. So for evolution to work, you must have a mechanism by which a change in the way the rules operate produces another pattern rather than something that simply fails. So if I take something like a watch and I pull a piece out or I change a piece, the watch simply fails. It's not evolvable. So it's absolutely key, the discovery that simply changing simple rules of interaction can cascade to very, very different patterns. Now, so this comes under the sort of the description of cellular automata, identical components. They call them cells. They could be our cells with DNA in them, each with an internal set of rules of interaction. In our case, it's the DNA or the genes coded by the DNA. The patterns emerge as the rules are applied iteratively. Growth turns out to be really important. If you don't have growth, you can't produce the complexity. It sounds stupid when you say it, and of course, we all grow, but we think of that as just something that happens. Well, if it wasn't for growth, we couldn't produce the sort of complexity that we see. And there's a twofold hierarchy required to produce these patterns, the cells and then the DNA within them. It really matters the way we're constructed. And so you've all seen patterns like this that can be generated with just a few lines of computer code. And remember, we have about 6 billion little pieces of information. So a tiny amount of information can do this. Imagine what our DNA can do. Well, we know what our DNA can do. It produces this. Brains and hands and blood vessels and kidneys. And some of them look more biological if you wiggle the parameters in the right way. And then, of course, there are things that are similar in the biological world. Antennae of a moth. The branching patterns of our lungs, and here, bronchial tubes, and then the capillaries or capillaries, depending on what part of the world you're from. Now, a lot of people emphasize that you can get complexity coming from these fractal type things, but there are other ways of producing organized shape without having fractal or chaotic processes. And so here, there's a way of making shells with 5 rules. So what happens is you start with an aperture. The animal's there, it deposits a little ring of shell behind it, and then it moves. And it deposits shell behind it as it does so. And so here, simply by changing the absolute size of the aperture, you can get a range of different shell forms. Now what you can do is have that aperture drop as it grows. And so you can get things like this to things like snails. 2 simple rules now, coherent changes between them. Or I can change the relative size of the aperture, or I can change the shape of the aperture, or I can change the orientation of the aperture. And so with 5 very simple rules, exercised through growth, I'm able to produce a huge range of shell forms. These are not chaotic, there's nothing to do with chaos or anything like that in it. It's quite simply an iterative process with rules. So all I have to do is change, say, how fast the animal grows with respect to growing of its shell, and I can change the morphology of its shell. And so here is a computer-generated shell. It's got fractal ornament, but it's got a non-fractal iterative process for the growth of the shell. So I don't want you to think that complexity arises simply through chaotic processes. Yeah?
So those shells on the previous slide those were all made on a computer?
They were all made on a computer with these 5 rules. Yeah, this is from Wolfram, who wrote the Mathematica, and this book called, what's it called? A New Kind of Science. It's about 2,000 or 3,000, 1,000 pages long. So now I want to talk a little bit about the DNA that we now begin to understand is responsible for these rules that actually operate in organisms rather than just relying on sort of computer simulations. But I think computer simulations, I tend not to think of computer simulations as valuable in trying to actually mimic what life does, but to try to explore fundamental ideas that are key in operating. And so here, you all perhaps know this story. How many know this story? Yeah, okay. So the standard story, who doesn't know the story? That's the key question. Yeah, okay. So there are a series of genes on the Drosophila chromosome that are arranged in order. We have about 4 sets of these, and they lay down the Cartesian coordinates of the body axis. So it takes the front to the back in the embryo and then into the adult. And in some sense it says there's going to be a front and there's going to be a back, and I'm going to divide it up into regions because I want different regions to build different structures. And there's another set of genes that does dorsal ventral, and there's another set that does left and right. You've got to create the space within which the organism is going to exist. And then, for example, on the second thoracic segment, there's a gene called ultrabithorax that makes the wings, and then there's a third segment, with ultrabithorax, this one here, that says if you have ultrabithorax, then your job is to make this thing called a haltere. And what it is, it's a modified wing, it's a little gyroscope, it spins at high speed. Have you ever noticed dragonflies fly? They're not particularly good at it. They've got 4 wings. Flies have converted that second state into this gyroscopic stabilizer, and so they can negotiate your food really well. So it turns out if you knock out the ultrabithorax gene, sometimes instead of getting 2-winged flies, you get 4-winged flies. I've not converted the haltere to a wing. What I've done is I've said, you are no longer the third thoracic segment, you are a second thoracic segment. So it's changed the identity of the third thoracic segment into the identity of the second. And so it builds second thoracic segment structures, which happen to be wings and this characteristic pattern of bristles. So we're a little bit like a computer program. There's something that calls up an algorithm that then uses other genes to make structures like wings. And so I think the wings are growing rather more like these cellular automata. And then we have master switches telling you which sets to go on. And so obviously we've got about 20,000 genes and that's 6 billion base pairs of information. Rather than being just one cellular automata, we can have one for kidneys, one for lungs, one for bone, one for muscle, one for brain, probably many for brain. And what you need then is a series of master switches to tell you which ones to switch on when and where. So we're actually collections of these cellular automatas rather than individual ones. But given that amount of information, I don't think that should come as a surprise. Here's another example of one of these switching genes. So there's a Drosophila head with, I'm too short. There's the eyes there, and then there's the antennae. There's a gene called Idolatry. You know, the way geneticists classically work is you knock a gene out. That's how you discover what it does. So you knock the gene out, you don't get eyes. That's why it's called eyeless. And so there, you can take that gene and inject it in the embryo where the antennae should be forming, and you can get eyes forming at the end of the antennae, or you can get them to form on the wing. So the wing would attach to the body here. There's the growing wing there, and there's an eye. So the ommatidia and the bristles and the pigments there, antenna and leg. So the leg would attach to the body here somewhere, and there's an eye there. They're not hooked to the brain, but they indicate the fact that we have these genes within it that switch on, they're master switching genes that switch on the cellular automata that are responsible for producing things like eyes. Now, one of the spectacular discoveries, particularly as a paleontologist, of this fundamental genetics is prior to the discovery of these genes, we all felt that different animals had to be built differently. We don't look like a sea urchin or a worm. The discovery, of course, is that most of these genes occur in all animals. So we're all different variants of the same set of rules. We're different variants of the same set of rules. And to give you one example of that, it turns out in humans we have a gene called PAX6, also in mouse, that's equivalent to the eyeless gene. And what you can do is you can take the mouse PAX6 and put it in the fly and ask, Mr. Fly, do you recognize it? It's like putting the PC disk into the Mac, which doesn't work, or the Mac disk into the PC, which does work. And what do you get? Drosophila, the fly, says, oh, I recognize that. I'm supposed to switch on the eye-making genes. And it does. It makes an eye. There's been at least 1 billion years of evolution between the last common ancestor of us and a fly. 1 billion years, with all those mutagenic effects happening, and yet the fly recognizes the mouse gene. It's unbelievable. So that modular, interchangeable nature even transcends species boundaries sometimes. So one of the genes in the Drosophila is called Tin Man. So what does Tin Man make? Yeah, Wizard of Oz, right? Tin Man has no heart, so it doesn't make a heart. So life encourages variety by reshuffling information. I just want to give you one empirical example. So back to our anterior-posterior patterning genes, the Cartesian coordinates. And if we just take two of those genes, they're called ultrabithorax and abdominal A, we can plot them on a series of crustaceans, and I apologize for this diagram. So Artemia, who knows Artemia? Sea monkeys, brine shrimp. Okay, so that's that one there. And then how about Homarus? Yeah, it's the lobster, right? Good. So we now know the three ones at the front are the head, and then that's the thorax. If these two genes are on, you get locomotory appendages, and when the genes are off, you get some other kind of appendage. So it looks like the patterns of appendages depend on where these genes are switched on. So there's a growing recognition now that most changes in morphology don't stem from new genes, but different combinations of rewiring the existing genes. So I had those growth of cells working out, we just changed the rules. Same sort of thing operating. Same battery of genes, you just rewire them differently, you get different morphologies out of it.
When you say rewire, you mean turn them on and off in different places or at different times?
Exactly. Rewired in the sense of it's really a metaphor, isn't it? Where one gene will act as a switch for another gene. So if that switch gets expressed earlier, then the target genes get switched earlier. And a lot of our development, we tend to think of our development as I make a kidney or a lung or a heart. A lot of developments actually co-involve with each other. So there are these blind crayfish that live in caves in the Midwest, and they have a rhodopsin gene, which is a light receptor. They don't see in the cave, it's pitch black. You'd expect the evolution of the rhodopsin gene to evolve very rapidly. It doesn't. There's no natural selection for light reception. Why is that gene evolving slow? Well, it turns out that rhodopsin is necessary for the formation of the optic cup, and the optic cup is necessary for the formation of the forebrain. And also rhodopsin happens to be useful for light reception. But you can't get rid of the gene, because without it you don't make the optic cup, which means you don't make the forebrain right. So that a lot of our development depends on these tissues interacting with each other. But back to the cellular automata now. Cell talks to cell, which tells which cell to be what when. And so by rewiring, what I mean is changing those rules by which the cells talk to each other or when they talk to each other. Yeah. Right, so that they are chemicals produced by a cell. You call them transcription factors. So the rule says that engrailed will switch on ultrabithorax, that's a rule. And then ultrabithorax will negatively regulate antennapedia, which will positively regulate some other gene.
So it's not a series of A's, T's, C's, and G's, it's actually...
Yeah.
Well, it has to be traced back to that eventually, right? You're saying you don't change the gene, you're changing the rules.
Yes, absolutely critical. So a lot of people study the evolution at the ATGC level. I don't think this is so important, unless changing the ATCs changes when a gene gets switched on or off. So the correct level of organization is not to understand the ATGCs, but the genes in their networks and how they control how cells talk to each other. So the question is, where does meaning come from? And what I'm arguing is the meaning in terms of making morphological form comes from the gene networks and how they're wired. So let me draw an analogy. If I want to understand a book, the ATGC level is almost like going in at the pixel level. Pixel, 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 oh, it's an A. Pixel, 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 oh, it's a T. Oh, at. What happened to the C? Cat. So pixels make letters, and letters make words, and words form sentences, and sentences form paragraphs, and the meaning comes at about that level, doesn't it? The sentence or paragraph level. So if I take the Bible and the Quran, I'm not going to understand the difference between those by going pixel, 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 pixel. I'm not going to understand the difference between a sea urchin and a human by going ATTTGC, ATATT. I'm just not going to do it. Those things are making genes, and those genes are talking to each other, and those genes are directing which way tissues are going, and we're made up of tissues. So you have to have a biology that talks about how the ATGCs translate into the way tissues talk to each other, which make organs, which make us. And I think we're still a fair way from doing that. So, long answer, but is that?
That's good.
Okay, good. So what drives biological complexity? And this one is particularly directed at the sort of the creationist types. Life builds from the bottom up, so we build up from the nutrients that we eat and the rest of it, but at some fundamental level, this is what drives life. It's the sun. For the last 4.5 billion years, we've been bathed in a remarkably steady flow of photons, some 84 trillion watts per day, pouring onto the surface of the Earth. Human consumption is about 2 trillion watts a day. It's an incredible amount of energy. And if you count them, it's about 20 trillion photons over the entire surface of the Earth per day. We are absolutely bathed in this stuff. We don't notice it very much because we take it for granted. Well, what does that stuff do? So I want to come to this rather tricky thing called the second law of thermodynamics, and we all get taught that it's about the world running down and life opposes it, and it drives me around the twist because there's another way of teaching it. The second law of thermodynamics basically states that when energy flows through a system, just the occasional photon coming through every day from our sun, you expect increased order and you expect material cycles. That is, the second law of thermodynamics basically says in energy flow situations, you expect the system to explore the improbable. What happens, the photon comes in, it forms a bond. That's improbable. You have a molecule form now. Eventually that bond breaks, releasing the energy. Life and death. I formed a molecule. Before it breaks down, another photon comes in and I join with another molecule. I've got a bigger one now. Always sustained by that pressure of energy. So it's almost like we're blown up from the energy of the sun, like the way you blow up a balloon. So that's what's driving the system. You turn the sun off and we disappear presto quick. So the key to the emergence of complexity, we've seen this before, simple components, this time carbon, nitrogen, oxygen, hydrogen, rather than cells, simple rules of interaction, the rules of chemistry, covalent and ionic bonds, leads to an incredible array of different molecules when you have energy flowing through the system. And you all know Miller's experiment where he took artificial lightning and shot it through what he guessed was the proto-atmosphere on the Earth, just 4 gases, including water vapor. And quite quickly he's able to form a large range of complex molecules. They form and act as precursors for the next one, which acts as precursors for the next one, and precursors for the next one, and the next one, and the next one. And of course sitting in there are a bunch of amino acids and alanine, building block of DNA, so the building blocks of life get produced incredibly easy. It's a long way from being life, don't get me wrong. But there's a case where thermodynamically this is what you expect to see. Increased order and material cycles. Now, I wouldn't say you would predict life for humanity if you went back to the Earth 4 billion years ago, but at least in principle, you should expect to see increased order happen. Where most people take the second law of thermodynamics to say, we should expect in principle things to fall apart. In an energy-driven situation, you shouldn't expect that. This is what you should expect. So we have to retrain our intuition and start teaching thermodynamics correctly, rather than in the mathematical way that it starts as if there's a closed system, there's no energy and matter entering it, then the entropy increases. Well, yeah, fine. And it's a dead system. And we happen to be a living system, so we need to adjust it for thermodynamics. So increased order in life is manifest. Material cycles dominate at all scales. We live, we die, we breathe in and we breathe out. We eat, we excrete. At the molecular level, almost every 2 weeks, virtually every protein in your body is replaced. Every protein, every 2 weeks, round and round and round and round and round and round. We are being driven at a phenomenally fast rate. We don't recognize it because it leaves a stable form. I recognize my hand day after day after day after day. So here's one of these invisible processes operating that's critical to evolution. So here's a diagram from the book showing, simultaneous creation and destruction happening within us all the time. So finally, we need more than this to explain how something like us arrives. Great complexity could arise, but why should it be as complex as we are? Why not stop at something simpler? And this last part is much more classically Darwinian. So what is it that shapes the biological complexity? What is it that creates? Yes?
Can you just go back a second
Yeah.
to your comment about this energy flow into the system and, you know, how that shows up where we're every 2 weeks, you know, creating new proteins. Doesn't that lead or leave open the option for absolute mistakes?
Yeah.
Because you've got all of these things happening over and over and over again. So every single time you have to redo it,
Yeah.
you reopen the option for a mistake.
Yeah, it's unbelievable, isn't it? I mean, now, for us,
How does that work?
well, it works. We have things called cancers. We have things called Alzheimer's disease. You get malignancies growing. So something went wrong, and now these cells, in the cancer, the cells forgets where it is. Oh, I'm a liver cell. I obey the cells, the instructions from my other cells. I forget. I just start to grow. So those things do happen. If I'm a plant, of course, as I grow my branch out, I then grow a fruit and a seed. Those mistakes then can get propagated to the next generation. That might be a bad thing, it might be a good thing. For us, it doesn't happen because we sequester our germ cells away early. So yeah, mistakes can happen.
So I'm wondering where the advantage is.
Oh, I'm not sure there is an advantage, it's what happens. But there's intense selective pressure to minimize that error rate. Because obviously if things go wrong at age 6 and I die, those errors don't get propagated onto the next generation. Variation followed by selection, Darwin. So the Darwinian process is responsible for the fact that we're so beautifully tuned that we can turn over our proteins every 2 weeks and still be utterly viable for up to 100 years.
I just wanted to say that I look at it also as a little bit of a balance, even if you have that rule applying. If you look at other things like communication in particular, signal transduction, a lot of it is not de novo synthesis, it's just regulating the enzymes that are already there.
Yes.
So I think.
But even the signal transduction pathways, each of those proteins have been constantly remade and remade and remade and remade and remade. So those are proteins, right? So all of those proteins they're not the same ones 2 weeks later. It's a little bit like, you know, you change your tires on a regular basis. We don't tend to change bits inside of our watches. I don't know enough of which cells replace at high rates versus low rates. Bone certainly remodels, but I don't know how fast that does. So let me press on with this, and I'll answer more questions at the end. What shapes biological complexity? So I think there's something very important here in terms of this issue of optimizing versus maximizing. It's very easy to think about maximizing wealth, power, glory. Of course, most maximization is akin to addiction. It means that other things get left behind. Organisms tend to optimize rather than maximize, and I want to try and explain that. And it's the optimization process itself that leads to complexity. I'm going to try and show you how that operates. And so I've coined a new principle. I'm calling the principle of frustration. It captures the notion that different needs will often have partially conflicting solutions. I can't both watch television and read well. I certainly can't sleep and read at all together. So that the overall optimal design for an organism, or the way you organize your day, will rarely be optimal for any of the specific tasks you need to perform. There are these trade-offs. You've got to decide where you're going to partition your resources at what particular time. And this has phenomenal consequences for the way morphology evolves. So here's a computer simulation done by someone at Cornell. And what he has 6 rules of making a plant. So there's 6 genes. There's the probability of branching on the left side and the right side, high or low. There's those angles that the daughter branches come from. And then there's the angles in this direction. So indicated here in those arrows. So these little dots here are the little tips of the branches there. 6 rules, 6 genes, really simple compared to the number of genes we have. He initiated evolution with the first land plant. It probably isn't. I've just discovered at the last national meeting, but it'll suffice. Here are 3 of those rules diagrammed for you instead of the 6. Can't draw 6 dimensions. Here's the simple land plant. And what he does is variation followed by selection. Change the rule a little bit, increase the branching rate, decrease the branching rate, increase the angle, and then select for some task and see whether or not it does better. So it's the Darwinian process through and through. If you select a reproductive success, Seeds occur at the end of branches, so you want lots of them, and you want them to disperse, so you get an upside-down broom. Doesn't look much like a tree. If you select for light interception, you want to have your branches out horizontally to collect as much light as possible, and you don't want overshadowing. And there are a couple of different ways of achieving that. Mechanical stability. Long branches horizontal break off, and that's a bad thing. So if you have long branches, they better be vertical. 3 optimal solutions, none of them look much like trees. These do. Now frustration sets in if you want to select for all 3 simultaneously. I can't both optimize light interception and reproductive success. This one's horrible with respect to mechanical stability. So what happens when frustration sets in? One solution? You get 7 solutions, and they become morphologically more complex. Now, when I present this often, giraffe's neck, natural selection for increased neck size. If we just teach that, you tend to fool people into thinking I should be able to look at the morphology and guess what the selective pressures are. All of these are roughly equally fit, and the morphologies each represent a compromise between light reception, reproductive success, and mechanical stability. I can't go to a real organism and tell you what the selective pressure is for producing this. There ain't one, there's usually multiple ones, and they're constrained by the rules by which the animal is constructed. So in some sense we do a disservice if we only teach the giraffe's neck. Now let's add a 4th need, minimizing surface area. On land, desiccation is a real problem. One of the things that kills burn victims, you lose your skin as you dry out. So that's going to minimize surface area as much as possible, which is going to fly in the conflict of reproductive success and light interception. 20 solutions now. Same genes, same developmental rules, just 4 needs. Frustration sets in. And everything with a letter on it happens to correspond to something we see in the fossil record by the end of the Devonian. And the Devonian is the time period in which plant evolution really gets going on planet Earth. So I'm able to mimic, or Carl Nicholas is able to mimic, plant evolution with 6 simple rules and 4 simple needs. Unbelievable. So it's frustration that's partly responsible for shaping. So yes, it's variation followed by selection, Darwinian style. That's fine, but it's even richer than that. So limitation is organizing. It's really spectacular. So we're optimal solutions. And I just want to point out that here's a simplified food web. Here's a description of 1,000 feeding links between 92 species in a lake in Wisconsin. A simplified food web of the Northwest Atlantic. There are intense selective pressures on all organisms most of the time. And frustration is going to prevent them from evolving very much. I might want to decrease my surface area, but that's going to decrease my light reception, so I can't do that. So I think stasis in the fossil record is because of frustration. Even in a changing environment, I can't change my strategy very much. And most of us know for our own schedules, our schedules don't change a lot depending whether it's school holidays or not school holidays, because the needs still have to be met. Frustration sets in. Time is relatively short, but you've all heard of the Cambrian explosion of animals. So I'm going to simplify a very complex anatomy and simply characterize it by these beasts that occur in the latest Precambrian and things like trilobites that occur in the Cambrian. And when I've been teaching this material, I often confess to my students I don't find these very interesting, because I love things that are intricate with knobs and bobs on them, full-rig sailing ships. And it's funny when you say things that simply, ideas suddenly strike. All of these fossils that occur below here have no eyes, they have no antennae, they have no setae, they have no claws, they have no armor, they have no bite marks. They don't have any organs of interaction with organisms of the same size. They're not interacting with each other. Where here, everything is characterized by tooth and claw, ways of interacting with the world. That's the fundamental innovation that occurred here, and I think what happened is the number of frustrated needs increase. Now I've got all these other species to worry about that I didn't. And so the same process that I showed happening with the plants, I think, is exactly what's driving the Cambrian explosion. And it's radically different from what most people argue, because most people want to say, I've got a complex animal versus a simple, I must have new genes in here. Developmental biology, which I can't go into now, suggests that those genes are actually already present here. And what's happening is the increase in frustration that's extracting out that combinatorial potential that's sitting within this genome. So I had that simple bunch of cells growing that produced a very checkerboard pattern that was really boring. I changed a couple of rules and I got the very, very complex pattern. The complex pattern is the one that's selected for because of frustration. Same rules in principle, just slightly different variants of it. Now I know there are gene duplications sitting in here, etc. There is an increase in complexity in the genome here, but I don't think that's what's fundamentally important. And so, trying to tie things up, the emergence of biological complexity flows from the hierarchical organizations of living beings, tissues, cells, DNA, given the richness of biological information, the fact that DNA encodes the rules, these are the genes and the gene networks, that govern the interactions between the cells, the fact that the application of those rules during growth may lead to complex form, and that when you have complex interactions between organisms, you enhance both the diversity and the richness of form by extracting out that rich potential. So it's kind of a little bit unsatisfying, but everything matters. It matters that there are lots of other species around. It matters that we're made up of tissues, which are made up of cells, which is made up of DNA. It matters that the rules of chemistry operate by protocols. It matters that genes operate by protocols. It matters that cells operate by protocols. And what's interesting about that is we're built from the bottom up and watches aren't. Organisms built hierarchically and watches aren't. We're built with interchangeable parts at the molecular level. Watches aren't. Parts combined via protocols, not in a watch. We're information rich, watches in comparison are not. And we grow, and watches don't. And what I've tried to show you is that all of these things conspire to make us both complex and evolvable. Oh, and the combinatorial, the different combinations, means that we're evolvable. And also this final point, which I just want to re-emphasize, the fundamental difference between watches and animals are largely invisible and only been discovered through the laborious efforts of science. Made of cells, the information rich, the encoding of the rules, and the complexity emerged when there's simple rules in growth. This is the computer science discovery. And I think that's important because people often come to you from a religious background or a family background where people around the dinner table are saying, look, we're built, we're a watch. You know, Dad says, I don't see how we can possibly evolve. And Dad isn't trying to convince his kid. He's just, that's what he honestly believes. And so it seems to me, and when I teach, you've got to honor what the kids, I mean, you know this, you've got to honor what the kids bring to the classroom, what the students are bringing to the classroom. And so I've constructed this talk in part to say, let's honor what their intuitions might be as honest junior inquirers of the world and try to convince them why some of their intuitions might not be right without belittling the fact, well, you're just dumb, you don't know this, or etc. So I'm trying to convince my Uncle Dennis that if he knew these things, he might not believe, he might actually believe in evolution. And in fact, over the last 3 or 4 months, I think I'm winning the war fairly nicely. So I've written this up. It's kind of a little bit of in a draft form. Is there an email list for this group? Yes. I'm happy when I've finished that the next week or two to email the manuscript of this. It obviously has poor illustration, but at least I've written the text of it. So some of this is there. So I apologize for not having that ahead of time. It's just I'm getting feedbacks from my friends, and some of them are pouncing on me for things that I should have got right. So I've got to fix them before I send it out. So as tempting as it is to view us as being like watches, we are fundamentally different, and I've tried to explore those ways. I said, I think this is a lot of fun. Works from ecology to myself. How many of you know this thing? Okay, it's really, I've only found it a couple of years ago. It's just magnificent.
Yeah, I have a question about the Cambrian explosion.
Yeah.
Over the summer, there was a book by Sean Carroll, where he claims that the Cambrian explosion he thinks was due to the origin of sort of control regions in the DNA that sort of make the rules more complex. And then I saw another book, which I haven't read yet, where the author claims, which is similar to what you said, he claims that the Cambrian explosion was due to the evolution of vision.
Oh, yes.
So it seems like one is working on the rules and the other is working on the interactions.
Right, so the vision is too simplistic in the sense that if you have no vision, there are plenty of ways of interacting with the world. So I think the vision is just grabbing too small a piece of the world, but it captures the fact that if I have a way of interacting with the world, vision, I think jaws make a big difference. I think predation is critically important. In terms of the new control regions, and this is very important theoretically, and which I've just written this up for a journal, is that you can't change if you don't change the wiring of the DNA. So I can compare all these species and they all will be wired slightly differently. So it's very easy to say, therefore, the reason why there was a Cambrian explosion is because the wiring changed. The wiring had to change to get different form, but why did it change? And so I think what's happening is that he's right. I think that modular system existed earlier than Sean thinks, and that the reason why the wiring changed is because we went from one or two needs to many, many frustrated needs. And so it became more important to find more complex forms to deal with all those needs. So to me, the causal force is ecology, the invention of ecology. It could only be achieved by rewiring the genes. So we have to know the way that rewiring operates, but it's necessary, but it isn't sufficient to explain what's going on with the Cambrian explosion. So philosophically, I've always been very opposed to these sort of two reductionist approaches to the scientific approach, or we can explain it by looking at the gene or the ATGCs. I just don't think it's going to work that way, and I'm trying to explain how. So,
are you saying that environmental pressures could cause a change in wiring almost purposefully?
no. So what's happening is things are rewiring all the time. We had the earlier question.
Yeah.
So rewiring is happening all the time. And so you're creating new mutants all the time. But if I'm an upside-down broom, any mutant that makes my branches go that way lowers them. I'm going to be less fit. That's going to get selected out of the population. So the rewiring is happening all the time, but the same ecological need, the variants get selected out, selected out, selected out. So the dominant force in evolution is stabilizing selection. The absolute dominant force. I mean, bugger this natural drift stuff. Dominant force is natural stabilizing selection at the morphological level. And then the world changes, and then if I need light as well as seeds, then doing that with my branches starts to matter. And then I've got to wait for those rewiring events to occur to get the branches to start doing something different and produce more of them so I can go out laterally and then make lignin stronger so my branches don't break off. So then the rate at which evolution happens is controlled by how long it takes for those innovations to appear. And they're relatively slow. We see them happening on geologic timescales. 5 or 10 million years, not 100,000 years or 10,000 years.
I read an article recently about the exposure of an organism to a certain set of environmental conditions actually immediately within one generation changing the wiring. It was like the mouse, the white mouse that made little brown mouse.
Uncle Dennis loves these arguments, yes.
But so, and that little brown mouse carried on that pattern through so many generations, and some experiments with Neurospora that could for 60 generations like an environmental impact didn't change the sequence in the DNA, but changed the expression of genes. How does that?
One of the discoveries, both theoretically and molecularly, that's happened, which is really important, is sometimes a small genetic change can have a large morphological change. And one thing which I have left out of this talk, Down syndrome, so an extra chromosome 21, right? On chromosome 21, there's a gene called ETS2. You take the ETST gene in mouse and give it an extra copy. Just one gene, there's no change in the gene, it's just upregulated a little bit. And the mice look like Down syndrome. Domed heads, short faces, malformed neck vertebrae, malformed. There's these systemic changes. So we're back to the fact that tissues influence tissues. So there's no innovation there at all, just increased regulation, and the whole morphology changes. So you can get tiny genetic changes that can have large ramifying effects, and you can have tiny genetic changes that have virtually no effects, and they're distributed in some fashion. A lot of these experiments where you get evolution happening almost instantaneously, my suspicion is that within the population, sometimes in the wild, that variation's already there cryptically. So does that?
I mean, Well, it was like these fat white mice, they fed the mother I can't remember, some amino acid free diet. And it influenced her progeny. It changed there were no extra genes or chromosomes, it didn't change the sequences.
I don't know the story well enough, but there's this thing called sort of genetic imprinting.
It was epigenetics.
Right.
And it was sort of like saying, well, maybe Lamarck was right in a twisted kind of way that acquired characteristics could be inherited.
This process called phenocopying, where you can not change the DNA, but if you starve something, then enough of the transcription factor doesn't get there, so those genes don't get switched on. And then eventually there seems to be you can actually get that encoded in the DNA. And I'm not sure how that mechanism operates. But there do seem to be some ways of feeding back into the DNA, which is not fully understood yet. I think it's really important. We need to be open to the fact that there are mechanisms that we might not yet have discovered, in terms of genomic type events. I don't know what they're going to look like yet, but you've got to be careful. It's funny when you're teaching, isn't it? Because you've got to teach them enough structure that they can get a good grade on their exams and assignments. But you don't want that structure to be so tight that it then confines their thinking to the way this is the way it must be or has to be. So you've got to give people structure first before they can relax structure. And I find that's one of the biggest dilemmas in teaching. I've got to teach concepts so they can start to get an intuition to what's happened, and then you start to unteach it. I've played an awful lot of chess. When you first start chess, they're very simple rules. A pawn gets a value of 1, a queen's 9, a rook is 5. Don't give it up. And then don't move pieces multiple times when you're opening the game, and make sure you occupy the center. You should see modern chess now. All of those rules are out the window. But you can't do that until you have the structure to understand what happens, and then you discover you can relax them sometimes. And I think at all levels of teaching you have this dilemma of teaching structure and information, and I find I'm often constrained by the need to grade. I mean, this is a fairly difficult talk. How on earth would I write an exam on this so that I could grade you guys fairly on whether you've comprehended it? I don't know how to do that, so I tend not to lecture like this when I'm lecturing to students because I'm constrained to assess them in some way. And I think that's partly just my deficiency as an assignment type person. But so I think the same thing operates. So what I'm saying is that I think there are mechanisms in the cell yet we don't understand. And phenocopying is one of those processes. No, I said no. Okay. What I said is that there's been 1 billion years of evolution. 500 million years down one side, 500 million years up the other.
It is just before the Cambrian.
Yeah, so sorry, but there's been 1 billion years of divergence of evolution. So that's...
So you're suggesting that the dominant force of teaching is stabilization too?
I'm tempted to get lured into a quick, snappy answer, which I won't comprehend. Say that again. Do you want to pursue that any further? Or? It's teaching. I find myself, how am I going to assess how my students are doing via some sort of criteria that's exportable? So what I do, of course, is I do bits of these. Some of this I do in my lectures. And so I've got bits that are easier to assess, sort of core information. And then I'll do bits like this, where I might not finish up asking any questions on it, I'll find a way of asking a question that's relatively general. Yeah, or is it hedging your bets type things? What can you rely on now versus long-term success? I came from a system that has fairly harsh grading. High distinctions, 1 or none each year, distinctions about 3 or 4%, credits, pass 1, pass 2, and most people get the pass 1, pass 2. And so, I would leave this sort of stuff. If you master this, you get the high distinction. And there are many years where no one will get a high distinction. And so there's a hierarchical level within the Australian system where just getting the basics down was fine. It was perfectly respectable. To get into the honours program in undergraduate, half of your second and third year courses had to be at the credit level. That's it. No distinctions, no high distinctions. In this country, I think there's a real problem with this grade inflation thing, which everybody's pushed up against that A, and so you don't have a possibility of sort of differentiating people on ability, but also I think commitment and interest. Most students who are exceptional, it's because they've got exceptional commitment and interest rather than just pure machinery. And so our system said it's okay for most of you most of the time to have no special commitment and interest, and you will not be penalized in any way. But there's occasionally some of you will have that, and I'm going to provide a mechanism by which I can reward that on paper by giving you that A, rather than most of the time, which most of you do, is get C's. So I topped my university. I got half A's, quarter B's, quarter C's. And I translated that into a grade point average when I came here for graduate school, and it came in as a grade point average of 3.2. It didn't look very competitive, but I was fairly naive about that. So, I mean, you all know about the grade inflation problem, but so I'm very glad for myself that I came from a system. Also, too, our system was much more forgiving. There are some subjects that I really hated, so I set a high school record in English of 38 straight zeros. I just wouldn't have made it this far in a system where I had to do well all the time in almost all things. I would have fallen behind almost immediately, and I know I would have been upset because I didn't feel I could be good at all things at all times, maximizing, and so I probably would have just given up and done something different with my life altogether. So I think this pressure early on in school is really detrimental as well. But that's just my own personal... Yeah, so the snowball... So Joe Kirschvink at Caltech is the first person to propose that there were these giant hard freezes not long before the Cambrian explosion. You get enough ice and snow forming and it reflects all this sunlight, and then you don't get enough heat to melt the ice, and the world just gets frozen hard. Volcanoes continue to happen, and they slowly build up CO2, and the atmosphere slowly warms, and eventually the ice melts again. And so this is called the snowball Earth. And then Paul Hoffman and Dan Schwag here sort of picked up on that. And their argument is, wow, it's an amazing, unique event in the history of life. It must have caused the Cambrian explosion. Geologists and paleontologists' primary way of knowing is by correlation. If I'm a carpenter, have a hammer, the world looks like a bunch of nails. If I'm an isotope geochemist, I can convert my view of the world into something I can shove through my machine. If I'm a molecular biologist, I've got to work out how to convert it into gene language. So a geologist has to look for things that occur at the same time in the geologic record. Correlation is not causation. And in fact, the theory that I developed here is partly developed in response to both Harvard and MIT workers in geology who want environmental change driving evolution. And my thought is, I don't understand how an environmental catastrophe can create new levels of organization. Why do you go from jellyfish-grade organisms to things like us and brachiopods and trilobites and clams by freezing the world hard? I mean, I just don't see it. So yes, it's a phenomenal, unique event. I just don't see how that can happen. And I realized we had no theory of biological complexity, organization. There is no theory. So in some respects, what I presented for you here is my first attempt at developing a theory of biological complexity. So in some sense, this has been an outcome of my frustration with the inadequacy of using environment to explain genuine evolutionary innovation. So that's my sense in that. Also, the damn things are too early, but that's another issue. Oh, Wolfram. It's called a New Kind of Science. I think it's 2002. It's utterly unreadable. It's got heaps of pictures in it. I've only looked at the pictures. Well, I shouldn't say that. Okay. Yeah. So there's a thing you've never heard of before. It's called Google. The complex food web is from the NOAA website, I believe. I haven't got it credited there. And then this one here is from, the previous one is from a group at University of Cal State, San Francisco, 1991 paper in Ecological Monographs. And that's readily available from their website. So Martinez, ND, 1991, or if you just do Martinez and Cal State, San Francisco, you'll find their website quickly and download it. I think it's very nice now. I'm on an editorial board of some of the review journals in our field, and we've just changed to a policy of actually just engraving on the images a reference, which means people can find it better, and I don't have to worry about misrepresenting stuff. And then this one, I think I just did a Google search, and I use this just pedagogically because you can really relate to that as a North American kid. You go to, so I find myself in my own mind giving blocks of time, usually in PowerPoint presentations. I still use overhead projectors when I lecture because I want to go at the same rate that they can write. I don't like using PowerPoint for material that they're going to be examined on. And then I reserve PowerPoints, which I do every sort of 15 or 20 minutes to break their concentration. I use those for stuff that is just way too complicated for them to write down. Harvard students are the type that want to write everything down, to try and expose them to things that are beyond the scope of the course. I think there's a real trap in teaching, particularly at the undergraduate level, of trying to present stuff that they're going to be responsible for entirely. And that limits the scope with which you can do. So that's some of the tricks that I use. Yeah.
So how do we get this kind of new complexity of genes and expression to get these complex organisms? Wouldn't you think that that would be somehow environmentally driven, due to change of environment is changing the simple rules that they're trying to function under? You see what I'm saying? If we have a massive environmental change, suddenly our tree might not be trying to go straight up, our tree is trying to go out. So we're changing some of the simple rules.
So I have a hierarchical view in my mind. So let me see if I can tease this apart. Is that in those computer-generated plants, I had the probabilities of branching and the angles. In some respects, every one of those plants, whether it was just a simple thing like this or something rather complicated, was just a different set of particular values for the branching rates and the angles. So in terms of sort of genomic complexity, they're all the same. They have a branching rate of P1, a branching rate of P2, an angle. They're all the same in terms of complexity. They're just tweaking the particular values. When I showed those cells that were dividing, I had a simple checkerboard, and then I had that really complicated one. They both have 8 rules, and each rule either says I'm either on or I'm off in the daughter products. So in terms of the complexity of the rules, there ain't no difference. No change in the complexity of the rules, and yet I can produce simple things and complex things. So as the environment changes, it might be that it selects for a different combination of the rules, which, if frustration is present, I think will increase complexity. So people have this very simple mapping. More complex means more complex machinery to make it. And what I'm trying to say is I can design a system that has simple rules where I simply tweak the little parameters inside it, the ones and the zeros. It doesn't look any different from a first principles point of view, but it looks very, very different in terms of outcome. So here, there's the set of rules, boring pattern. Change the rules in a minor way, more interesting pattern. Change it again, very rich pattern. But in terms of the rules, look, there's just 8 of them. And the only thing I'm tweaking is whether or not, if the left, I don't know, left from right. Left versus right. The 2 left are on, the right one's off, it's on. So does that make...
I guess so. I guess I'm just trying to tack down what decides the changes of rules.
So the rules are changing all the time. Variation happens.
So when you get more rules changing, like an environmental change or interaction between different new creatures in your environment, you've got more rule changes.
I would say that when you change the environment, you may select for a change in which rules, but it won't usually fundamentally change the complexity. In terms of the Cambrian explosion, what changes the level of complexity is the number of frustrated needs that have to be met. But most of the time that doesn't happen. So most of the time if you get some environmental change, you can't change because you can't have shorter branches because that's going to mean less light. But sometimes it will. At the back and then there.
Based on this concept of rules, and you would expect things to get less complex just as often as they get more complex.
Only if the number of needs they have to satisfy gets less. It's a very good question. When things become parasitic, they get less complex. The super parasite of all time, of course, is the virus. It's cheating, of course. It's borrowing everybody, all your machinery. But it's a very good question. But I think what happens, remember our food webs, the world's a really intense place. But what you might expect is if you go to a small island where the number of species drops, you might expect simplification to occur. So that's an interesting, actually that would be an interesting test of the idea. So there's an increase in oxygen through this time geologically. And so I don't know enough geochemistry to know, but I suspect the big freezes might have something to do with changing the world's chemistry so that you get enough oxygen in the atmosphere that might have been permissive of the evolution of large animals. So it may be that these deep freezes are important in terms of changing the way our world has operated fundamentally. We go from about 1% present oxygen to about 10%. And that makes a fairly big difference in the maximum body size you can have. And if you're really, really tiny, 100 microns, predation isn't going to be the same as it is if you can get to a meter. So that's I find relatively easy. And so one of my colleagues has argued, for example, that the dinosaurs become extinct and then all the mammals take over. So you have a massive level of innovation. So maybe what happened is the snowball Earth called a mass extinction and then the Cambrian explosion took off. It's too early. But there's also some carbon isotopic anomalies at the Precambrian boundary, and maybe that killed off the simple ones. If you compare mammals to their nearest relatives at the same level on a cladogram, on a diagram, those things are the diapsid reptiles. And the diapsid reptiles include dinosaurs, pterosaurs, crocodiles, ichthyosaurs, plesiosaurs, placodont's. Those are the main groups. You have things in the ocean, you have things in the air, you have giant things like dinosaurs. The diapsids get hit hard, a lot of them go extinct, and then what happens is the synapsids, the mammals, take over and they produce things that are large in the air, in the oceans. In terms of biological organization, we're not much different from the diapsids. So there's, I just got two clades, two evolutionary lineages. One gets hammered at the NKT and the other one then reoccupies those niches. And because our biology is slightly different, the animals look slightly different. So from my point of view, all I see is a sort of a large-scale ecological replacement of the reptiles, these extinct things, with the mammals. But I don't see it as a fundamental change in the level of organization of the biosphere. If you're mammal-centric, then yeah, it makes a huge difference. So most mass extinctions are characterized by a lack of major innovation. Sometimes ecologically it happens. Before the Permian mass extinction, most species are non-motile. They're attached to the substrate in the ocean. After the Permian mass extinctions, a higher proportion are mobile. We don't understand that at all. It's an empirical record. Ecology fundamentally changes, and I've got a colleague who's just retired from Virginia Polytechnic who wrote this brilliant paper in '77 called Seafood Through Time. You could live off seafood today easily, right? Clams, snails, lobsters, sea urchins, you're great. You go back to the Cambrian, oh, it'd be miserable. Tiny little brachiopods, tiny little echinoderms. There are no clams of any decent size. There are no snails. It'd be horrible. It'd be hard to sustain something of our body size, quite frankly. So there have been long-term secular changes in ecology in that way. Sometimes it looks like the mass extinctions can restructure the way ecologies re-emerge. We have virtually no understanding of what causes that. Just don't know. Right, so I'm currently writing a paper now where that's one of the possibilities. So I've got 4 or 5 phases of evolution where I can increase, what I can do is I can leave the number of boxes the same and increase the level of frustration and go from simple organization to complex. Or what I can do is add an extra box. Or maybe what I can do, instead of having 2 colors, I can have 3 colors. That increases the dimension. And so what I've been doing is exploring the different fundamental modes in which evolution will occur. And I think I've got them classified well enough that you give me a morphological change, origin of birds, origin of whales, extinction of trilobites, origin of Cambrian explosion, and I can tell you which of those major variants is operating. So my argument is, for the Cambrian explosion, I don't need fundamental genomic innovation to get the explosion. I think it was also occurring. Because as you say, you get the chicken and the egg thing. If I now want to be more complex, adding an extra rule now might make a difference. And so, for example, we've got 4 sets of the Hox genes, not 1. The Hox genes pattern the outside of the animal. They're a gene duplicate of something called the parahox, which patterns the stomach. If you duplicate it, then you can leave the stomach doing exactly what it should do. Nothing should change there. But now you can have the duplicate set, the Hox genes, pattern the outside of the organism, making a complex array of appendages. So that gene duplication is clearly really important in terms of differentiating our exterior. Those duplications might have been happening all the time, But since there was no selective pressures to make your exterior complex, they just disappeared again. So I agree, it's going to be this sort of cyclic type thing. So yeah, good catch. So I think the biphasic life strategy of marine invertebrates is to break frustration. C3, C4 plants. Stomata, water out, CO2 in. Go to a dry environment, I'm in trouble, water going out. C4 plants, I can separate those processes, I can move into an arid environment. I think what happens in Homo sapiens and why we're so dominant is we are the masters of breaking frustration. If I'm an individual species, I have to do everything myself, catch my food, etc. But what do? I've got lip balm. I've got a comb, if I can get it out. I've got a pocket knife. Can't find it. There's a pocket knife. It's got multiple blades on it. Put my pen's over there. I've got a microphone, a pair of shoes. Go to a kitchen. Every task, I've got a specific tool which has minimal frustration. Only has to satisfy one task. We can hunt mosquitoes and whales quite comfortably. So our technological capacity means that we can utterly break frustration. We can make a machine and a device that's task specific. It doesn't have to be strong like a screwdriver so that it flexes on my head. So I can allow it to be highly deficient for all other purposes by making bucket loads of them. So I think what makes us really special is we can break frustration in a spectacular way. So we can perform an incredible range of operations in the natural world that an individual species can't. So for example, penguins and orcs and puffins, where do they occur? They only occur in cold upwelling areas. What's special about cold upwelling areas? There's lots of nutrient in the water. If you're a bird, you fly, you can have rare resource. Catch a mouse there, a rabbit over there. You can't give away flight unless there's a rich resource. If you go into water, you're giving away flight most of the time. So you can't be flighted underwater like a penguin unless the ocean is incredibly rich biotically. So you can't both be a penguin and morphologically and fly, given the aerodynamics of water versus air. Frustration. I can't both live in the water and survive off it and survive in the air. Doesn't affect us. You build a boat, you go fish lots of fish. You farm wheat on the land. So that's what makes us, in some sense, really special and what makes us ecologically so dangerous. Because our numbers have gone up and we can operate in all ecosystems, or many ecosystems, really, really well. So I'm trying to write this up in a more general sense, and the last theme is going to be humans and frustration, or our capacity to circumvent it. But I think one of the things that evolution tries to do is to break frustration if it can. And one of the ways of doing it is behavior, changing your developmental mode, etc. So that's a good question. So I think with that, I'll end.